hacer un pequeño parrafito de Mechanotransduction, pero tú conoces. Si yo no tengo miedo. No voy a decir una palabra.
Buenos días. Muchísimas gracias por acompañarnos aquí en el auditorio del Yeliscali en este evento. Muchas gracias también a la gente que nos está viendo en redes sociales y a quienes en su momento vean las grabaciones en YouTube. Muchas gracias por acompañarnos. Bien, para comenzar, lo primero que tenemos que revisar son algunas medidas de seguridad. Por favor, en caso de que se tenga alguna emergencia, las dos salidas de emergencia que tenemos están en los costados de la parte superior del auditorio. En caso de que se requiera ayuda o cualquier necesidad que tengan, por favor diríjanse con quienes tengan una playera como esta o quienes porten un gafete parecido a este. Estamos para apoyarles. Bien, nos acompañan en el Presidium el doctor Víctor Manuel Velázquez Aguilar, director de la Facultad de Ciencias, la doctora Tatiana Fiordelicio Col y también el doctor Pierre-Henri Puecho. Les damos la bienvenida, muchísimas gracias. Bien, Bien, a la doctora Tatiana y al doctor Pierre los presentarán más adelante en sus propias presentaciones. Muchísimas gracias por acompañarnos. Bien, esta semana en el Symposium Internacional de Mechanotransducción Celular y Tecnología Biomimética vamos a tener a varias de las personas con mayor conocimiento, renombre y experiencia a nivel mundial en el ámbito de la mecanotransducción celular. Nos estarán presentando por medio de algunas pláticas el estado del arte actual en temas de investigación y desarrollo. Tendremos además varias sesiones experimentales donde podrán ustedes de primera mano adentrarse en el desarrollo de plataformas microfluídicas y biomiméticas. En un mundo cambiante, la adaptación es esencial para la supervivencia. Y en un mundo de egos e individualismo, la colaboración y la cooperación es una revolución. Para hablar de mecanotransducción celular necesitamos de eso. Por un lado, la física nos dirá cómo es la interacción y la química nos enseñará sobre los canales de comunicación celular. Todo dirigido por la biología, quien entiende ese comportamiento. Pero ahí no termina todo. Ese conocimiento es requerido por la medicina, quien sabe que esos medios son el origen o la consecuencia de algo a nivel orgánico o sistemático, que se debe atender. Y es ahí donde las ingenierías y el diseño nos otorgan tecnología biomimética útil, funcional y ergonómica, para que luego las áreas administrativas y comerciales las hagan llegar hasta donde deben de ser utilizadas. Toda esta cadena no es trivial y esta semana revisaremos los primeros eslabones y los expertos nos acompañarán, nos mostrarán los diversos esfuerzos que a nivel global están realizando. Así que por favor preparen su agenda. Esperamos que este simposio internacional sea el primero de muchos en el que podemos compartir la trans y la multidisciplina, porque en el Ansbiodit es posible. Le cedemos la palabra a la doctora Fiordelicio. Último.
Bueno, como siempre no somos ejemplo de puntualidad, entonces empezamos sin que estén todos los que están registrados, ni modo. Y empezamos porque además la agenda está internacional y entonces tenemos muchas intervenciones en Zoom y si se van retrasando, para los participantes resulta complejo conectarse. Entonces vamos a iniciar. Pero me gustaría retomar lo que súper bien dijo Raúl. El equipo de Ansbiodit, que está compuesto por más de 30 profesionistas, que son físicos, matemáticos, biólogos, médicos, y ahora estamos trabajando incluso con arquitectos que tienen desarrollo industrial, pero también con ingenieros, es decir, con muchas áreas. Y como decimos en el Ansbiodit, solamente aportar conocimiento tiene que ver con la participación de todas las ideas, tiene que ver con la comunicación de todas las ciencias, de todas las materias, y tiene que ver con trabajar juntos para lograr aportar algo hacia la sociedad, no solo en el individualismo, yo hago mi artículo y ya, sino en el colectivo, es decir, como colectivo de pensantes que somos, como colectivo de profesionistas que hemos tenido el privilegio de poder estudiar en esta universidad. ¿Qué le aportamos a esta sociedad? ¿Qué le damos a esta sociedad? Y eso es parte de lo que en las Biolit queremos hacer. ¿Y qué le damos a esta sociedad? Puede ser en muchos ámbitos. Por ejemplo, en un simposio de mecanotransducción, en el cual queremos que los alumnos empiecen a ir más allá, se salgan de la cajita y digan, vamos a juntar la física, la biología, la medicina, pero también en otras cosas como los productos médicos que hacemos, como la instrumentación médica que hacemos y muchas otras cosas que los invitamos a conocer en Enlace Biobit, en las páginas de YouTube, en las redes y hoy en la tarde en las sesiones prácticas. Muchas gracias por estar aquí, por participar, por registrarse. ¿Y ahora?
Hola a todas y todos. Me voy a cambiar un poquito de lenguaje para que es más fácil para mí. Thanks you a lot for being here. And so this symposium is in fact the second one. We did one with Mathieu, the founder of the Land Biodit, very long ago in 2018, where we started all this kind of very nice collaboration between France and Mexico. And this is what we keep on. It's what you will see with the different people who will speak. It's at the same time physicists doing biology, biologists doing physics, and the exchanges, as you all mentioned, between the two. This is what is very important. This is a very dynamic community that has been lasting now from the last, I would say, 25 years. For the main mechanobiology, it started much more long ago, if you look at the papers, in the 70s or even before. So thank you a lot for being here. It's in the frame of this CNRS UNAM connection that we started the project, and I hope that we will be able to enlarge the community, enlarge the communication between different universities, different labs, different people. And thank you a lot for being here. It's a real pleasure for meeting you again all here in UNAM. Muchas gracias a todos y todas.
Muchas gracias. Quiero agradecerle a la doctora Tatiana Fiordelicio y a Pierre Henry por haberme invitado a esta inauguración de este evento, de este simposio, que me parece muy importante porque realmente está en la frontera del conocimiento. Para este simposio se registraron 80 personas, dentro de las cuales 15 presentarán sus investigaciones. Son ponentes de Francia, Estados Unidos, Canadá, Holanda y, claro, de México. Me gustaría dirigir algunas palabras a los estudiantes porque, por una parte, además de que los ponentes que se han registrado van a intercambiar conocimientos, para esta facultad es muy importante que nuestros estudiantes reciban ese conocimiento. Creo que es una de las áreas más excitantes que tenemos en la facultad, de hecho que se desarrolla en este laboratorio nacional que dirige la doctora Tatiana. Y a mí me hace recordar ciertas cosas. Por ejemplo, si ustedes se acuerdan, hay una paradoja en la computación en la cual nos decían que la velocidad de cómputo, obviamente en las computadoras, dependía del número de transistores que podía albergar un chip, ¿sí? Entonces, cada vez tienen más. La paradoja consiste en que ¿hasta dónde vamos a llegar? ¿Cuántos transistores puede uno construir dentro de un chip? Dicen que es posible que lleguemos incluso a nivel molecular, incluso atómico. Algo parecido está sucediendo en la biología. Están viendo, ahora están investigando qué es lo que sucede dentro de la célula. Ahora hay que reestudiar si realmente la mecánica como la conocemos funciona a ese nivel. Seguramente tiene algunas variaciones. Los fluidos, la microfluídica, seguramente tiene que recomponerse las ecuaciones que las gobiernan. Es decir, de alguna manera la termodinámica a nivel mesoscópico tiene otro sentido. Así como la constante de Planck en mecánica cuántica o la constante de Bohr a continuar en esa dirección, porque ya vieron, esto es un esfuerzo multitransdisciplinario. Aprovechelo y muchas gracias. Pues empecemos con este interesante congreso. Muchas gracias a todas y todos.
Queremos especialmente darle las gracias a la comitiva francesa que está aquí. Son 3 investigadores que hicieron evidentemente el esfuerzo de estar con nosotros para compartir no solo la parte teórica, sino la parte práctica, y al doctor Jacobs, que también vino especialmente desde California para estar con nosotros, lo cual nos alegra mucho que hayan decidido hacer el esfuerzo de venir hasta acá. No es fácil para nadie en estos momentos viajar, dejar las cosas que tenemos en nuestros países. Entonces, especialmente darles las gracias por estar aquí. I know that you didn't understand my Spanish, but I'm just saying thank you so much for being here, to do the effort of being here. We are really, really happy to have you, not just in the talks, but also in the practice part. Thank you so much.
Yeah. Yeah, and then because he will present you, and he has like just one slide to present you, and then we choose each to your.
Okay.
Solo porque todos usan Sorry.
Oh, no,
It's just that we are used to use the drive, but of course, when the internet is just slow, then we have problems too. Parte and microphone is not enough. Okay, done.
So, thank you all for being here. And I'm going to present Professor Jay Groves. He's a professor from UC Berkeley. He has revolutionized the study of cell membranes and signal transduction with the innovative patented pattern supported membrane technology that has been patented. He works integrating physics, chemistry, and biology to explore how spatial organization and dynamic molecular interactions drive cellular signaling processes. This is very important for the symposium that we are organizing here. And he is leading a diverse lab in physics, chemistry, and biology and engineering, which is also very important for the transdisciplinary work that is needed for this kind of research. And he's going to present to us his talk right now.
Y ahora ponemos este aquí. Yeah, this one.
Should I use this microphone or is there something?
This is.
Yeah. Okay.
And then you have one. Here you have to, yeah, it's
That's good.
point.
That's good. Yeah.
And then we are all seeing this one.
Yeah, so let's do this.
Great, thanks.
Okay. Over a little bit.
You're gonna need more of?
No, I think this will be fine.
Okay. May I put this? It's for the streaming.
Yes. Great, thank you. Well, it's really a pleasure to be here. It's my first time to come to this university. In fact, it's the first time I think I've been to any university in Mexico. This is a good place to start, probably for a visit. Also, I was very excited to get the invitation because, you know, California, we feel we have somewhat of an affinity with Mexico, and finding more ways to interact, I think, is a really positive thing. Okay, so my talk today, I'm using the title Imaging the Mechanics of T-cell Signaling at the Membrane. And I would say the theme has been, throughout my career, has been physical, more so than mechanics specifically. But we've always been taking a physical approach and a physical interpretation of these things. But that certainly includes a mechanical aspect. Now, the system we've been looking at is the T-cell immunological synapse. And it's kind of, I started out as a physicist doing soft matter physics in a physical chemistry department with very little
biology, actually, but I was always interested in moving into biology. And it was the year that I was applying for my job at UC Berkeley that the immunological synapse really was debuted in the literature. And I didn't know anything about T cells or immunology. I just saw it. And I thought, this is a terrific system for someone who has an interest in physics to go after cell biology. And there was no more thinking than that. But now, 25 years later, I find myself still working on these things. And what you're looking at in the background, it's actually a really beautiful light sheet, 3D kind of microscopy movie taken by Eric Betzig. And the T cell is the one that's shown in red here. And it's interacting with an antigen-presenting cell. Now, what it's doing is it's scanning for antigen. So these would be peptide fragments that are displayed in display proteins on the antigen-presenting cell surface. And the bottom line is the T cell is identifying viral infections and cancer and clearing those cells from your body. So when you get the flu or COVID, the reason you recover is your T cells are the ones that really saved you in the end. But when you look at this, it's this incredibly dynamic interface that's also extremely active. You can really see that it's not just two cells that are passively adhering to each other. There's a lot of active movement in here as well. And all of this is part of the molecular recognition process. So there's a lot of interesting molecular physics going on in these systems. And T cells are also very important in health. And there's been really an exponential growth in developing them as therapeutics, targets of therapeutic drugs, and even engineering T cells themselves for therapeutic purposes. And the 2018 Nobel Prize went to one of my former colleagues, Jim Allison and Tasuku Honjo. But Jim was at Berkeley when he originally did his work on the checkpoint inhibitors, which many people would say gave birth to the cancer immunotherapy field. We don't work on this at all. We're a very physical and extremely academic lab, but I show it just to remind people that it is actually important that we understand T cells. And it's not just the biology. Understanding the physics of them also contributes to this field. And I think that's an important connection. Okay, so as a physicist or a physical chemist looking at the T cell, I see it really in a different way than the medical scientists looking at it, I think. And in particular, one of the things that's amazing about the T cell is that they're single molecule sensors. So T cells can respond to one or a handful, 3, 4, or 5 individual molecules. It's important that they're able to do this because this is what allows your immune system to detect very quickly a growing viral infection and ramp itself up to clear it fast enough to save you. So there's been evolutionary pressure to make T cells as sensitive as possible. But T cells are also extremely precise. They don't trigger on 0. And from a physics background, many of us know that it's not difficult to make a sensitive detector. I started out doing single photon spectroscopy. You can always turn up the gain and see single photons, but then you're amplifying noise as well, and you'll see a lot of noise and occasionally a real photon, but you can't tell them apart. So that the amplification isn't difficult, but to avoid amplifying noise and still be single molecule is extremely challenging. And T cells do manage to do this. It's very easy to show it. So for decades, we've known T cells are operating in this space. But how they do it is really presents a big problem. And here's how I would phrase the problem is that if from the physics side, if we needed to make a very sensitive and precise detector, the first thing you think of is I need to get good components, good high quality, low noise components to build this thing. But when we look at a T cell, it is not built from precise components. In fact, the molecular components that make up its detection pathway are incredibly noisy. And this is very easy to show, and especially when you get down to single molecule imaging experiments like I will show you, you can see all of the background noise that exists. I find it amusing even that the biological field has a name for it. It's called basal level signaling, as if, and it may even have a functional purpose. And certainly biologists have interpreted it that way. But the physics view of this is the basal level signaling sounds like noise. And it sounds like it would not be a good thing for T cells to be single molecule sensors. And so this is one of the, I'd say, big physical problems in the T cell is how do you build a single molecule sensing system out of noisy components? And how did a biological system find a solution to that very challenging problem? Okay, so we're going to have to get into a little bit of the details about how the T cell signaling system works to explain, I think, the experiments we're going to be doing next. So I'm just going to give a quick background on this. And so I mentioned the T cell is using this T cell receptor to recognize peptide fragments that are displayed in a protein. In mouse, it's called MHC, HLA in humans. And these proteins are displaying basically a snapshot of all the protein production that's happening in a cell. So if a cell is infected by a virus, you're going to have peptides from the viral genome showing up on the cell surface. And those peptides don't exist in your genome. And so you have T cell receptors that will recognize them. And if any of these things are seen, then what you know that there's something foreign being reproduced inside that cell, and you must destroy it as quickly as possible. Similarly, with cancer, you'll get peptide fragments that were not part of the native genome, and T cells clear those as well. So they play a major role in preventing us from getting cancer. In fact, we're all getting cancer all the time, and it's not cancer until your immune system starts to miss it. That's the only time you would actually develop any disease or even have it stay in the body. But that's all happening at this recognition event here. And it's an interesting way the T cell does this as well. So the T cell receptor binds this MHC, and it's scanning for peptides that it might recognize. So it has to bind a lot of ligands, a lot of different ligands, and discriminate among them. It doesn't just have one that it binds and that activates it. And so the T cell receptor, unlike a lot of receptors in the body, it's not triggered by binding alone. The ligand needs to bind, and then it needs to stay there for some extended period of time. So T cell receptor is a timing measurement device. It measures how long something is bound. And if it's longer than a certain amount of time, some kind of cutoff, that will activate it. And this process is called kinetic proofreading. So the T cell receptor is one of the classical examples of kinetic proofreading being used in biology. Okay, and so these things have been known for decades. This is kind of the basis of the adaptive immune response. Now, when a T cell receptor gets triggered, what that really means is that a series of kinase proteins get recruited and activated, and these start to phosphorylate other things in the pathway, namely this protein called LAT, linker for activation of T cells. It receives these phosphorylation events, it gets phosphorylated itself, then it recruits a whole series of other signaling molecules, which then transduce a signal downstream and ultimately activate the T cell. So this is sort of the way we would describe how the T cell signaling pathway is laid out. Now, what's missing in this sort of description, first of all, it's just totally non-quantitative, right? I've just sort of in words said all the players in this game and what they're doing. But it's really impossible for us to put any kind of quantitation to how this functions and how does it actually achieve single molecule sensitivity and how does it avoid being activated by noise. There's a lot more that we need to do to really understand that. And what happened back around the time I started at Berkeley 25 years ago now is that several different immunology groups began really for the first time to start to image that interface between the T cell and an antigen presenting cell. Before that, people thought of cell biology as doing bulk measurements. You'd grow up 1 million cells, you'd lyse them, and you'd measure how much protein of different types. And you'd see this average of a whole population. And back then, it was the beginning of starting to image exactly what are all these molecules doing, and particularly how are they moving around in space. And this is kind of how that I entered the field without much biological background. I was coming from a background in soft matter physics and microfabrication, actually. We heard a little bit about chip manufacturing before. And so I was a graduate student at Stanford, and I knew a lot of people in applied physics who were building their own chips for various things in the micro lab, fabricating them themselves. We started getting the idea, what if we could use tools from the microfabrication industry to design experiments that we might apply to cell biology, like physically interface a cell with a chip in a way that we could manipulate it. And so this was the sort of technology we were using at that time. We'd have a living cell on one side, and then we'd make a phantom cell consisting of a patterned surface. In this case, it's just very finely patterned metal lines on glass. But then we could assemble bilayers on this substrate, lipid bilayers, and they would form these single bilayer coating that was fluid on the surface, so things would diffuse around freely. It would look like a live cell surface to another cell. But we could also embed it with these fixed structures. And in these experiments, they're just static. It's not even electrified or anything, though you could do that as well. But here they're just confining the way these molecules move. So the first real experiment we got done on T cells was this one. And this was the image of what the original immunological synapse looked like. That's what was discovered around 1999. And that's the T cell, and it's sitting, in this case, on one of these supported bilayers. And you would see the same thing if you looked at a T cell sitting on an antigen presenting cell. It would form this bullseye pattern of the different proteins in that signaling pathway. And the question at that time was whether that spatial pattern had something to do with the signal. And way back then, this was an interesting question because there were a lot of people, and many of them would not admit it today, but there were a lot of people that believed you could fully characterize a cell if you could just list the copy number of each protein and its state, whether it was phosphorylated or not. And that you did not need to know anything about spatial organization, that just the contents list would be sufficient. And so we introduced this idea that we named the spatial mutation. And these cells were all chemically identical, so there's no chemical difference between them at all. But by allowing the cell to try to form one of these patterns on different pattern substrates, the constraint from the substrate would force the cell to pattern in a different way. So parallel line constraints like this, it would give a stripe pattern that was perpendicular to them like that. On grids of various sizes, we could partition it into discrete sort of mini synapses. And we had a whole slew of patterns like this. And then we would look and see, what are the cells doing? Are they signaling? And how are they signaling? And basically, what we found was this pattern is not required to activate the cell. In fact, it seems to be more of deactivating process than activating, because when we would disrupt it with these sorts of patterns, the cells couldn't turn off signaling. They would be stuck in an on state. So what was actually happening is cells were triggering before this pattern even formed. And then as it formed, what you were watching is the shutdown process. Now, shutdown of signaling certainly correlates with signaling because you don't shut it down unless you've activated. But the pattern itself was not the signal. It was something that came after the signal. So that's something we learned from that. But we continued to take these experiments further and also taking advantage of things that were developing in the microfab industry. So as computer chips were pushing towards faster and faster speeds, what was driving this was the ability to pattern metal and semiconductors on smaller and smaller length scales. And so we were just taking advantage of those advancing tools to make smaller and smaller patterns, getting down. These are 500 nanometer grids. The grids themselves are probably 40 nanometers wide. We're partitioning here on a sort of a 500 nanometer patch. This is a fairly small length scale to be able to control what molecules are doing in a living cell. And as we were doing these experiments, we also began to pay more attention to the fact that T cells were single molecule sensors and try to look to find that activation threshold, that dilute down the ligands that would trigger them and figure out, can we really see them activating on 1 molecule or 10? What is it? And use these sorts of structured experiments to explore if something about how the molecules organized spatially was important. Like maybe not one molecule, maybe it's not 1 molecule that activates a T cell. Perhaps it's 10 or 4. And we were at that threshold. We ended up doing these experiments and finding that it wasn't really a copy number of molecules. It appeared that the cell needed to see a group of 4 molecules that come together. That's what it appeared in these bulk experiments. And we could see that because if we had a certain density of ligands and we partitioned it so small that on average, it became very unlikely that you would ever have 4 molecules of ligand in one of these corrals. Then when we look at, here's like 100 cells and each one is a calcium trace, looking at whether it activated by tracing its calcium flux. Basically, this is not activating. This is exactly the same density of molecules here. Those are activating and they also are activating here. So we drew this conclusion and okay, we figured it out. We've identified that this group of 4 is the activator. And had we not been a quantitative lab, we might have stopped there and been happy with ourselves. But we are a quantitative lab and we looked progressively more deeply into this problem. And what we had detected in that experiment is evidence for cooperation. How many molecules could be near to each other in space for the cell's ability to see these things. But if we start to really think about the kinetics, by now we're starting to measure what is the kinetic on rate for binding of a ligand to T cell receptor. Actually, it's exceptionally slow. Much slower than most other receptors, in fact. And it has a kind of a long, medium-long dwell time. So once it's bound, it may stay bound for 10, 20, 30 seconds. It unbinds, but the chance of rebinding is very, very low. So then when we do the math here, this group of 4 molecules that seems like they could come together and at least correlated with activation, but they would never be bound at the same time. What they really are doing, the duty cycle between bound and unbound is far in the favor of being unbound. So you just have one bound and then it would unbind and then another one would bind a little while later. That's really what is happening there. And so how could they possibly know that they're in a group of 4 if they're never even bound at the same time? And is it even possible for cooperativity to exist through time rather than through space? So these are very physical questions in terms of how the T cell system works that basically we decided we could never answer using these bulk averaged experiments. And so just to clarify, what I would call a bulk averaged experiment is that even though we're patterning and looking at single cells, we're not visualizing the binding events. We're just putting a certain density of ligand, and we know on average, it's going to have somewhere between 1 and 2 molecules per corral at that partition size. And then we assume it's randomly distributed, and we look at thousands of cells and we average it together. That's a bulk averaged experiment. What I decided, though, about 15 years ago is that, did I do that? Not sure. I didn't think we would ever be able to figure this out using these types of experiments. And so we switched entirely to doing single molecule imaging. And what's great about the T-cell system is, since it is a near single molecule sensor, we can easily visualize all of the molecules in the experiment, all of the ligands to the T-cell receptor. In fact, what's visualized here is kind of an unusually high density of ligands, which we made just for the purpose of taking that picture. T-cells will actually activate with significantly less than this, where you only see 2 or 3 molecules in the whole image. So it's easy to see them all. And then what we'll do is we're imaging the ligands in one channel here, and then we'll image whatever we want inside of the T-cell to try to figure out how it's responding to these ligands. But now we're going to be able to do it not just cell by cell, but molecule by molecule. So we'll see how each receptor triggers. Now, this type of imaging this is live cell imaging that we're doing, but it's related to super resolution. Whenever you're looking at single molecules, of course, you see a diffracted image of the molecule, but you can localize it to a much higher spatial precision than the diffraction limit. So we're doing optical microscopy in live cells, but we do have about 50 nanometer spatial resolution in terms of seeing the individual molecules. And so that gives us a really very finely resolved window into the signaling pathway. Okay. There was another bonus that we found with this system, and that is when these ligands are in the membrane and free, they undergo free Brownian motion, and you can see them diffusing around. And this is a track of one of those molecules. When we image them, you just see them diffusing, and the blue is tracing its position. So it followed this trajectory all around. And then right here, something changed. That molecule was diffusing at a particular diffusion coefficient relatively fast. And then abruptly, it changes to a much slower mobility. And we realized that those abrupt changes in mobility result when that ligand binds to the receptor. The receptors on the T cell, the receptor is moving very slowly. The ligand's moving fast. And as soon as it binds the receptor, it slows down and it moves at the speed of the receptor. That was an important thing for us because now we can detect the binding event itself. So when does the ligand bind to the receptor and how long does it stay bound? And there were certain tricks we could play to make all of this more efficient. One of them is, we can track, if we do fast streaming, 20-second exposures in the microscope, we can visualize all of the molecules and all their Brownian motion. But if we lower the power on the laser and longer exposures, like 500 milliseconds, then all of the freely diffusing molecules just kind of blur out in the background. And what you see as well-resolved spots are only the bound complexes. So this kind of stealing an old photographer's trick of using long exposures to blur out all the fast motion, and then you resolve just things that are kind of fixed. But that works in these single molecule experiments. And then we end up with this view. And what I like about it as a physical scientist, it's a literal view of what is happening in that cartoon I showed in the beginning about the T-cell signaling pathway. There's the T-cell receptor. This is its ligand. And at this moment in time, a ligand bound to that receptor, and I saw it. And I can see it stay bound. In this case, it stays bound for a little over 30 seconds, and then it abruptly unbinds. And we can watch every single ligand in the cell, in the experiment, doing this. And so what we see is under the footprint of the cell, we see binding events. And they're flashing on and off. Each one is a ligand binding to a receptor, staying bound for a while. And we can see how the receptor and the ligand move together also. But this is a literal visualization of the process that we were imagining when I showed the first diagram of how the T cell signaling system works. And what we'd like to do is then go and image things that are happening inside of the cell around these binding events and see if we can directly image the propagation of a signal through the cell and basically watch the cell make a decision about whether or not to activate. All right, so the first set of experiments we did was kind of just a logical test experiment. And so there are these binding events. This is the same signaling pathway I showed before. It's just depicted a little differently here with the molecules drawn out, roughly what their sizes are, roughly. But if the T cell receptor is successfully activated, and you propagate a signal all the way through this pathway, one of the things that happens is this transcription factor, the nuclear factor for activation of T cells, or NFAT, gets translocated from the cytosol to the nucleus. And that's one of the definitive activating steps in a T cell. So when that happens, a T cell is in the process of activating. And it's a little bit off the screen here, but you can easily visualize this. You can see on the side screens there that when NFAT is in the nucleus, it's very central like this. And when it's in the cytosol, it's clearly peripheral. You can see over there. And basically, this is non-activated, this is activated. So that's the shift. And so that's something we could easily see. So the experiment to do was to visualize all of these binding events and simultaneously watch this transcription factor and see whether it's going to move from the cytosol to the nucleus. And when it does, that cell has made a decision. So we're basically watching the input and then watching the output of a decision, and we just want to correlate them to each other. So we did this, and a lot of things come out of it. It's a very complex set of data. But here's one key thing that was surprising. Now, when we think about things like binding events in terms of bulk chemistry, we teach people about kinetic off rates. And you can imagine thousands of receptors bind, and then if they all just sit there and wait, you'll have this unbinding curve, and you can calculate what it is and get an exponential shape. But when you do it one molecule at a time, it's totally discrete. The molecule is either bound or unbound. There's no slow decay of anything. But it's the probability of unbinding that's changing. And if you histogram up the observed dwell times, then they will map these exponential decays that we see from bulk interpretation. But we see it as a discrete series of events, and we can actually see that in the cells. Now, what we found is there's this decades-old understanding that T cells respond to ligands that bind for at least 3 seconds. That's an experimental observation from bulk experiments. So ligands that have, on average, 3 seconds or longer binding time correlate with cells that activate. The interesting thing we did in the experiment, though, is we could ask the question, well, what if a cell experienced 10 binding events, and they were all 5 seconds or 10 seconds? They were all longer than this, but not really long. Do those cells activate or not? And this experiment showed us that those cells did not activate. So although the literature is full of measurements of 3 seconds as a cutoff, we find cells that see 3-second events don't activate. They never do, in fact. What does activate cells are really long dwelling events. If there's one of those, that cell activates for sure. So then how do we put these two things together? And this is kind of at this interface of bulk versus stochastic or specific single molecule interpretation. Just because the average is 3 doesn't mean every binding event is 3 seconds. So a molecule that has an average 3-second binding time will actually have an exponentially distributed array of actual binding times, some of which go all the way out to 30 seconds. And what we observed is using these weak ligands is that we look at many, many cells, most of them not activating at all, and they're all experiencing binding events like this. But the rare cell that did activate always had that rare binding event. And so the T-cell system has actually set its tuning point to look at the tail end of a distribution. It's not even looking at the average. That was, to me, was very interesting because you have to try to think about how we do science. There's this built-in assumption that we all have to think about, that we often correlate average with average with average, assuming that the average is the thing that matters. And here's an example where it really isn't. Now, this is also a problem of signal transduction. And so I have this example. There's this running race in San Francisco. It's called Beta Breakers. It's actually the largest running race in the world, so I'm told. About 100,000 people run it every year. And if you ever go to San Francisco in spring, you should try to see it. And most of them will be running it wearing costumes of all sorts, some of them pushing grocery carts. There's a lot of partying on the way. That's all part of the race. I saw people carrying a tiki hut once, about a dozen. And they'll do this literally for the whole 7-mile race. So it takes all day for most people. But there is a small collection of about 50 or 100 people who are serious runners who actually run the race to try to get a good time. They finish it in 30 minutes, 35 minutes, whereas the rest of the people are rolling across the finish line 8 hours later. So the average time to finish this race is many hours. But the first people cross the finish line in 30, 35 minutes. And you ask, if the race starts at a certain time, and all you want to know is, did it start? And you're sitting at the finish line, you know it started as soon as the first few people cross the finish line. You don't have to wait for the average person to cross to know that. And that's kind of what's happening here, is the averages don't matter. As soon as the first few events happen, or you can be at the tail end of a distribution, but that can pass the information that's required for the cell to move on to the next step. So none of these processes in signaling require the average time or even the average copy number to occur, because it's an information propagation. And this really changes, I think, the way we think about doing experiments. Okay, but we still have no idea about mechanism here. We just observed a basic correlation. So if I try to go a little bit deeper into this, and we look again at that signaling pathway, right between the T-cell receptor and this calcium activation and the nuclear NFAT translocation, there is that phosphorylation of LAT, which leads to this assembly of other signaling and scaffold molecules, including critical ones like the RAS activator SOS and PLC gamma, which ultimately activates the calcium pathway. All of these things get recruited into this cross-linked network during the signaling process, and it has recently been discovered that this forms by a type of phase transition. So this thing, this little assembly here, is considered one of these biomolecular condensates. So it's a phase separation process of some sort. And it's happening right in the middle of the signaling pathway. So we went back to the imaging experiments and now decided to focus on this molecule, the LAT, inside the T cell. So we're going to image the binding of ligand to receptor, just like we did in the first pass at this. But now we'll see what LAT is doing to see if we can see these condensates. So they've been predicted, but never observed in cells in relation to a binding like this. So we do those experiments, and it's kind of hard to see on the main screen, but I think you can see on the monitors better. So what we're looking at is in red, it's the LAT protein in the T cell. And this is a highly zoomed in image. The T cell is probably about that large, is maybe a quarter of it. And the LAT is initially fairly uniformly distributed in the T cell membrane. And what we're going to see is one receptor bind one ligand. And right now there's 0. But if I advance a few frames through this movie, there's the first binding event. So there's one ligand bound to a T cell receptor and nothing else happening all around it. So that's a convenient experiment for us because we can watch what that one T cell receptor does entirely by itself. And as time goes by, eventually we can start to see, you can see there's some red accumulating underneath that green spot. So the green spot is the one ligand bound to a T cell receptor, it's diffraction limited. Underneath, we're starting to see enrichment in LAT. That's more than one LAT molecule, actually, dozens probably at this point. And that grows over time. And even you can see some movement between the receptor and the LAT as more and more LAT molecules grow into that assembly. And it keeps going, and eventually it dissipates. And if I can play it, that's almost a real-time bind, wait, always a wait, and then condensation process. And so this is what that T cell signaling pathway looks like for each receptor. And what we could do in these sorts of experiments is visualize every receptor binding event that the cell experiences, and then track the LAT condensation on each receptor. And what we find is that they're characterized, maybe I have to point it out over here, they're characterized by, always characterized by, here's the binding time, that's when that receptor bound, a long delay, tens of seconds, then an abrupt condensation, which ultimately dissipates. So each condensate on each receptor is doing roughly that. The delay time is pretty broadly distributed, though. It can be spread out. It can be quite long or quite short. But the growth of this, once it triggers, is very fast. So a very discrete condensation process. We also can observe, so this is just tracking these 7 binding events. This is what they all looked like. You can see the condensate forming in each case, and a couple of them fail. So in fact, a significant number fail. It's just because the ligand bound a receptor, even for a long period of time, doesn't mean that that receptor is going to successfully signal. What we see is it's actually quite binary. You either get a condensate or you don't. And if you get one, the condensates are all the same. There's no measurable distinction between them. So there's no such thing as binding for a longer time, getting a bigger condensate. No, does not do that. It's completely digitized at this point in how that signal is propagating. Okay, this was just zooming in on the condensate, the delay time. So I mentioned that they're broadly distributed. The average is 25 seconds for this delay, but some of them can condense within 3 to 5 seconds. So it's possible for a receptor to trigger that quickly and give a condensate. Now we can actually learn a few things from this, and I won't drag you through all the physics of it, but what it boils down to is the shapes of these distributions inform us about what type of physical phase transition could be happening. It's very asymmetrical shape, and it's weighted towards early times. And what that boils down to is a type of kinetic phase transition. So those of you that might have learned something about the protein condensates in the nucleus, there's a lot of discussion about miscibility phase separation or liquid-liquid phase coexistence. That's not what this one is. This is very interesting and distinctly different type of physical phase transition that's based on kinetic competition between two enzymatic reactions. One are the activated kinases on the T cell receptor that are phosphorylating LET, and the other are phosphatases that are competing with that and dephosphorylating. And this type of kinetic competition gives rise to these sorts of shapes. And the interesting thing about it is this, if you wanted to make a single molecule sensor that was suppressing noise, this type of phase transition is a very good choice because it has the tendency of erasing history. You'd never accumulate noise. The system is always going back to zero. But it takes a certain type of nucleation event to trigger it, but it doesn't accumulate any information. A miscibility type phase transition does. It is actually accumulating something before it transitions. So our speculation, or our hypothesis here, which I think is likely true, is that you needed to have this type of phase transition to make a T cell a single molecule sensor. Okay, so I can show you just a few more things. I mentioned that this is a really discrete process and that the amount of time a receptor stays bound to a ligand does not influence the size or value of the LAC condensate. But this is what that data looks like. So here's 100 successful binding events. So some of them are very short, all the way up to hundreds of seconds. That's how long the ligand stayed bound. This is the lifetime and size of the resulting condensate for each of those binding events. Now, what you can see very clearly is that there's no correlation here. If it gives you a condensate, it's just the same size as the long ones. It's no smaller. That means that's the end of the antigen discrimination process. At that point, the cell is committed and has no more information about the original molecule. It has successfully triggered a signal. What is different, though, between these short and long events is the probability that you get a condensate at all. So again, I think we have to look on the monitors on the side. So here's about 1,200 individual binding events that we tracked. What you might be able to see if your eyes are sharp is that, I can't see it, but I know it's there. What we've done is we've plotted this bar in red indicating the ligand binding time, and we've switched it to green if and when a LAC condensate forms. And so the traces that switch over to green, that's a condensation event. So that's a successful binding event. The ones that stay pink, the ligand unbound before you ever got a condensate and no signal has propagated from that T cell receptor at all. And it's mostly pink up here. It's very rare that those short binding events trigger a condensate, but it gets progressively more likely for the longer ones. And this is a map of that probability function. So this is the dwell time, how long the ligand stayed bound on the T cell receptor. And this is the probability that that T cell receptor triggers a LAC condensate. So I was proud of this. As a physical scientist, this is a real measurement of the antigen discrimination process at the level of a single receptor. So watching how a ligand triggers the receptor. And you can go and quantify these things and map them back to classic theories of kinetic proofreading. And I won't go through any of the details, except to point out this over on the monitor here. This gray curve is our measured kinetic proofreading function from that data. These yellow and orange are what are predicted by classic theories. And so you can see they fit for the first 10 seconds or so, but they become dramatically wrong after that. So we're missing an awful lot about how the signaling system actually functions with those oversimplified ways of looking at it. And so there's nothing wrong with them in principle. I think, but we need to realize that they don't answer the problem. They start it. It's a very first order approximation. But with new experiments, we can see much deeper into what the cells are actually doing. And there can be important consequences there. What it literally means that this function goes down to 0 is that if you fail to activate a T cell receptor after about 30 seconds, you've got no chance of ever activating that receptor. It's just dead. So some receptors just won't trigger. Why this matters? And I actually do some consulting in industry on these sorts of things with companies developing therapeutic synthetic T cell receptor activators. It's probably a really bad idea to make a synthetic TCR activator that has a very strong binding to the T cell receptor, because it's going to bind. And if it fails to trigger for whatever reason, and we know it's a low probability, look, this curve saturates below 30%, but it will never trigger. Holding on longer is just a waste of effort. The natural ligand doesn't hold on. It lets go and eventually rebinds. And it's that perpetual rebinding that just keeps trying, and activates it. So that's probably an important detail about how T cell system works. Okay, so now, in some of our more recent work, and this is not even published yet, though it is out on the bioRxiv, we took it a step further. Instead of looking at just the LAC condensate, we wanted to see if we could see signaling activity coming from those condensates. But just because we saw it form doesn't mean it's actually active. We'd like to see, can we detect the signal coming out of it? So we did this experiment where we would image, it's a 3-channel imaging experiment, looking at the binding events, the condensates, and then monitoring intracellular calcium levels. And the result that came from this was amazing, to me at least. What we observed is that almost as soon as we see it form, the condensate is signaling active and it contributes a single calcium spike to the T cell. And you can see these things here. So what's being plotted are in gray are the copy number of LAC condensates that we observe, and blue is calcium. Look at how these spikes line up with the LAC condensates. So the calcium spiking pattern that we observe in T cells is actually the stochastic sequence of LAC condensation events as they occur. We've actually been able to quantify this. We measured the impulse response function. It has no ringing, no delay, no oscillations. There's no oscillation at all in the T cell calcium response. Instead, it's just a faithful reflection of exactly the LAC condensation events. This time pattern in calcium signaling has been observed for decades, but it was always assumed to stem from some type of ion channel oscillation, like other cell types that do have very distinct oscillations, not in T cells. And when we think about it, there's a good reason for this. If you wanted to build a single molecule sensor, or if you're a physicist and you ever did things like single photon detection, it's very easy to get your electronics to have ringing and oscillation after photons hit. You're never going to detect single events that way. You have to overdamp the system so that when one photon hits, you get a sharp and strongly damped signal coming out of it. That's the only way to build a real single particle detector. T cell has managed to do that. And it seems to have tuned calcium signaling in a distinct way in T cells, differently from certain other cell types, to be strongly overdamped and to give these very well-resolved calcium spikes that are associated with each LAC condensation event. I do have a movie of that. I'll give it a play. It might look better on the monitors. So here it is live. So binding in the left, LAC condensates in the middle, and you see this pulse in calcium in the T cell, and that's just a direct reflection of the condensation events that are underlying it. Okay, let's skip a couple of cells here. And then to kind of sum this up a bit, you know, I would say our hypothesis is that this LAT protein condensation phase transition, it's playing a role in T cells, and that it is the noise filtering mechanism that's allowing T cells to be single molecule sensitive. The mechanism of the phase transition itself, this unusual kinetic phase transition, has the proper, always returning the system within a matter of seconds to zero. So it doesn't accumulate noise. It hears noise and it either triggers or it doesn't, but it doesn't build it up. And this is the way a lot of digital communication works. We use digitized signals so that you can ignore small noise and just make it a binary readout. The condensation appears to be doing that on the one end of signaling to provide noise filtering, but it also provides amplification. And it's again the same thing we see in digital electronics, that you have a digital signal, you use a very high voltage difference, relatively speaking, between the two states. So there's just no doubt about it when the signal is on. Same thing. So the LAT condensate, we were able to measure how many LAT molecules are in them. It's hundreds, 300, 400, 500 LAT molecules from one receptor. That's how many molecules are in those condensates. And that's enough, apparently, to trigger a cell-wide calcium spike. So the fact that it's a condensate and it has this phase transition, the phase transition makes it kind of hard to trigger. But once it triggers, it grows rapidly, and it gives the amplification that would be needed to detect those single events. And then what it appears is that the whole readout system in the cell, that is the calcium response, is tuned to be overdamped in such a way that these incoming pulses produce discrete spikes, but that they don't ring, they don't repeat themselves. And then it gives a fairly good measure, molecule by molecule, of how many ligands the cell is interacting with. Okay, so I see I'm at about 55 minutes. I have another 40 slides on how, which I was not planning to present. I just had them there just in case. I think what I'd like to do is stop here, and happy to ask questions. Don't be disturbed by the large number of slides shifting by. I'll put up the names of all the people who've been involved over the years, and happy to take any questions if there are any at this point. Thank you. Yeah. I can repeat your question too.
Right. I was just wondering if you could comment on, actually I had two questions. One about if you could comment on how the condensate actually happens, like physically. I guess when one says condensates, one is thinking that the lipid actually is looking like, I mean, I thought you wrote gel to liquid phase transition. So
Okay.
how should we think about it?
So let me answer that one
Okay.
first. That's a big question. So how should we think about these condensates? So if I can show that slide again, this one. What you could see in this depiction is that as these molecules are recruited to phosphorylated sites on the protein, GRIB2 binding to a phosphorylated site on LAT, and there's multiple phosphorylation sites. GRIB2 recruits SOS, there's multivalency. And this is just one of the multivalent bridges. There are a couple of distinct ones. It can cross-link to another LAT molecule. And this can just keep going, cross-link, cross-link, cross-link. And you get this kind of bond percolation network. So the phase transition is that. It's a bond percolation transition. And physicists will and do argue that it's not it's a different kind of phase transition altogether. It might be continuous, but nonetheless, it corresponds to a discrete transition. Now, one of the things that contributes to its sudden onset and rapid growth is that when this starts to assemble, there's positive feedback. Positive feedback on the kinase phosphatase competitive reaction, because as it condenses, it excludes phosphatases. So what you have is before condensation, you have a handful, 3, 4, 5 lone kinases on a T cell receptor competing with a sea of phosphatases. And they actually don't compete very well. They're phosphorylating and it's immediately being dephosphorylated, but it fluctuates. And if it fluctuates enough that you get the first few assembly bonds forming here, then the positive feedback kicks in, starts to reinforce it, and then it grows to 1,000 molecules very quickly. And that is what would be known as a kinetic phase transition. It doesn't exist without this positive feedback that changes the balance between a competitive kinetic reaction. It does not resemble a miscibility phase transition. Now, is there any miscibility going on here? I think the answer is yes. And I would argue that the decision is made here. It's the proteins. But these proteins are interacting with lipids, and there are distinct interactions there. And they almost certainly, almost certainly are driving lipid miscibility phase separation underneath it.
Do you think there's a feedback?
Could be. And we've measured that in a very crude experiment, but we've measured proteins that don't interact with these do get recruited into the condensate, probably because of changes that are happening in the lipids. And that's an idea that, as you know well, has been around for a long time. And I think it absolutely is happening here. But the distinction, I think the new layer that the protein condensate adds to it is how did you decide to do it in the first place? And that's the connection, yeah.
Okay, so the second, hopefully short question is, do you have any idea of how the calcium gets damped down?
Oh yeah, great question. Right, so we observed and are trying currently to publish a very controversial point that calcium is overdamped in T cells. Why does it, how does it get damped down? And so I don't have a good answer other than this. I don't think it doesn't need to be anything different from the expected calcium pathway. And we've done a lot of kind of classic characterization to see where is the calcium coming from. It is in fact extracellular, not ER based, and et cetera. But have this balance between triggering the receptors, calcium influx, and then activating a second set of channels. And depending on the concentrations, numbers of channels, the amount of volume in the cell, that's what determines whether it oscillates or it's overdamped. And so it's not like you have to change anything. It just tune the system slightly differently. And one thing T cell has is it has very small cytoplasm, very little ER calcium. And so you completely deplete the ER right away and immediately go to extracellular. I think these differences are probably what shift the exact same pathway that oscillates in one cell to being overdamped in the T cell. Yeah. Yeah. Yeah.
Just keep on with the calcium. Basically, I had two questions. The first one, is there a delay between the last condensate aberration?
Yeah, but we say it's basically as short. It's sort of unmeasurable, so it's 10 seconds or less, but not zero.
In the case of an extracellular one, you see co-localized spikes, spatially speaking.
We've never seen spatially co-localized calcium, but that might be a feature of the way we're imaging it, because it would diffuse quite quickly.
That could be a way to dampen.
Okay. Yeah, interesting point. In fact, I think that that is certainly part of it, is how quickly the cell depletes.
Because if locally you have a super high spike of calcium,
Yeah, there's a limit to how big you can get, for sure.
it's a volume to concentration to size of the zone where the calcium is that probably governs the damping.
Yeah, that's a very interesting point. Something to think about. Okay.
Okay, thank you very much. That's all the time we have for right now. So if you
One of these waters I was drinking.
No, gracias.
Bueno, ahora es el turno del doctor Alexander Boone. Es un investigador y académico cuyo trabajo ha sido pionero en el campo de la mecanobiología. En el campo de la mecanobiología se centra en la comprensión de los mecanismos moleculares y celulares mediante los cuales las fuerzas mecánicas influyen en la biología. Actualmente es profesor en la Universidad de Stanford, donde lidera un laboratorio que combina enfoques de biofísica, bioquímica e ingeniería para explorar cómo las células detectan, procesan y responden a las fuerzas físicas. Muchas gracias, doctor. Deseo la palabra. Thank you very much for being here with us.
So sorry because we have late.
Well,
You're
thank you.
sorry for being late.
Yep. So, hopefully you all can hear and see me. Is that true?
just give us.
Sure thing. Sí.
¿No? ¿Eso está bien?
¿No?
Aquí está. No. Okay.
So can you hear me now?
Yes.
Okay,
Yes,
great.
we can figure it out.
Okay, wonderful. All right,
But-
I will begin my presentation then, if that's okay. All right. First, I'd like to thank the organizers very much indeed for the opportunity to present some of our work at this conference. I regret very much that I can't be there in person. I am teaching this quarter, and my university has a strong attachment to the idea that I fulfill my duties. So, very much enjoyed the previous talk. And as you'll see, there are really cool parallels between the work that Jay Gross presented and some of the ideas that I'll explore over the next 45 minutes or so. All right. So the question that fascinates my group is one that I think many of you will be interested in as well, which is how do organisms build themselves? Now, we know from data from paleontology that the basic body plan of animals has not changed in over 600 million years. Actually, it's probably closer to 750. So here's a fossil sponge, for example, from the Ediacaran. And we know from evolutionary and molecular biology data that the same set of molecules that was used to construct that fossil sponge is used to construct all of the astounding and beautiful diversity of animal life that we see surrounding us. So this mouse, and you and I, the same set of molecules, broadly speaking, are used to construct cells as diverse as the cardiomyocytes in your heart that pump blood, and the neuronal synapses in your brain that together lead to the emergent property of consciousness. And so the question really is, how do you build a thinking, living creature, entity, starting from molecules? And of course, answering this question is going to be the work of, many thousands of scientists over many years. So my group contributes to this problem by thinking about how you bridge specifically from molecules to higher order assemblies that can mediate cellular self-assembly. So we've, over the years, used single molecule biophysical approaches to study how force is involved in this process, and I'll talk a little bit about that today. We're increasingly involved in cryo-electron tomography so that we can visualize this molecular self-assembly process directly. And then we've done and continue to do work at higher length scales. So for instance, work that should be coming out any day now, looking at the reproduction of the morphogen gradients that pattern development in the early embryo, and then a work we published some years ago looking at the physics that underlie the most important and basic processes in tissue morphogenesis. For today, I want to focus on the epithelial sheet. And the reason for this is that a sheet of cells with a top and a bottom, and as I'll talk about in the second part of the talk, a front and a back, is the basic building block of multicellular animal life. Once you have a sheet like this, you've defined the principal axes of any tissue. And if you roll this sheet up into a tube, you have the basic architecture of almost all of our internal organs. So think about the kidney, digestive system, blood vessels. Now, given its importance, it's useful to think about how you build such a polarized structure. In the first part of the talk, I'll approach this from a very biophysical perspective. In the second part, we'll think about it more from a signaling perspective. So what I want to focus on right now is the molecular players that build this apical, meaning top to bottom, meaning basal, polarized cell-cell junction. In vertebrates, this cell-cell junction is vertically stratified. At the top, you have what are called tight junctions. These provide a watertight seal between neighboring cells. And then below them, you have what are called adherens junctions. These are cadherin-mediated linkages between cells that provide mechanical robustness between cells. Without these two classes of adhesions and without their proper spatial organization, it is not possible to build a multicellular animal life. For that reason, we're quite interested in how this intricate structure assembles itself in living systems. Now, sharpening that question up a little bit, what I'd like to talk about in the next few minutes is what mechanisms underlie the intricate self-organization of cell-cell junctions. Now, some time ago, we found that the components in the cadherins-based adherens junctions are mechanically sensitive. What I mean by that is that the protein that links cadherins, shown here in blue, to actin has a counterintuitive but extremely useful force-sensitive property, meaning that its binding to actin is weak in the absence of force, but as soon as you begin to pull, the binding lifetime increases by about an order of magnitude. These forces, which we'll talk about in a little bit, are consistent with the forces that molecules experience in vivo. Now, there are several consequences to this. In these earlier studies, what we explored for the most part was the idea that this force sensing can make living tissues robust to external mechanical loads. So the idea is that if you have two cells that are attached to each other, they begin to experience some external force, that these connections will automatically become stronger to keep the tissue from ripping apart. And this is necessary for any animal that likes to move around or interact with its physical environment. What I'd like to do today is extend our thinking a little bit beyond that. Now, this is coupled to an observation that a recently graduated student from my group made, which is that there's been extensive investigation of just a few molecules that link one class of adhesions to actin, whereas the vast panoply of molecular linkages that are also necessary to build an animal remain unexamined in many cases, a biochemical level, and in all cases, a biophysical level. So what Vipul decided to do was to chip away at this deep lack of knowledge, this black hole in our understanding of how cells construct themselves. And in particular, to develop a strategy for examining the mechanical sensitivity of all these other linkages. Now, in considering this problem, what Vipul noticed is that many of the linkages between cell adhesion complexes, for instance, these components of the tight junction, and actin are mediated by a common scaffolding domain called the PDZ domain. Now, PDZ domains have been extensively studied from a systems biology standpoint. They are small folded domains that commonly bind to the C-terminus of their partner proteins. There are over 200 of them in the human genome, and they, among other things, commonly participate in the construction of condensates. Just for example, PSD95 and discs large are both important components of the neuronal synapse. We'll be talking about this protein zonulocludins 1 a lot in the next few minutes. Now, the other feature of PDZ domains is that their binding to their ligands is in general promiscuous. So here are some of the connections that occur at cell-cell junctions between a bunch of cell adhesion molecules and just two scaffolding proteins, ZO1 and L-afadin, or I'll refer to it as afadin from now on. And you can see that the PDZ domains in these proteins bind to multiple ligands. Each ligand in general binds to multiple PDZs. And I've in fact shown you only a tiny fraction of the really dense network of interactions that can occur at cellular junctions. Now, I would like then to focus on two questions. First, how do you derive order from such a complex network of interactions? Remember, I've told you that this vertical stratification is essential to building anything that has more than one cell in it, at least in human biology. Second, and possibly coupled to that, what Vipul noticed is that the response of any PDZ ligand bond to mechanical load was unknown prior to his study. So Vipul decided to examine both of those questions. Now, for context, I need to tell you just a little bit about the proteins that he chose to focus on. Vipul is going to focus on PDZ domains from two multi-domain scaffolding proteins that link cell adhesion proteins to F-actin. The first of these is called ZO1. It has 3 PDZ domains in addition to a whole bunch of other domains, and it links both directly and indirectly to F-actin. The second is a protein called ephadin. Ephadin has just one PDZ domain. It also binds directly and indirectly to actin. Now, both of these proteins are essential for proper embryogenesis and the organization of cell-cell junctions. Ephadin has quite prominent roles in cancer biology. And just for example, the ephadin and nectin connection is required for the assembly of the intestinal barrier and proper kidney development. Now, as a reminder, Whipple's fundamental curiosity was how these PDZ to cell adhesion linkages respond to force. But before he could examine that, he had to do some very basic biochemistry. So what we're going to look at as a model system for this question is the linkage between the C-terminal peptides of two cell adhesion molecules, nectin-1 and JAM-A, with the sole PDZ from ephadin. Nectin-1 is part of the adherence junction. JAM-A is part of the tight junction. We can see that these two C-terminal peptides bind to the ephadin PDZ with almost identical dissociation constants. And I have to say that these are necessary, but just about the least inspiring data that one could imagine measuring. This is a very normal KD for a PDZ domain, and there are no surprises here. What he did next is to examine if either of these PDZ domains might have interesting, or peptides might have an interesting force-sensitive interaction with the ephadin PDZ. To do that, Whipple made one long protein construct in which the PDZ domain from ephadin is linked by a long flexible linker to a biotinylation site, and then the nectin intracellular domain. Now, this construct folds back on itself. The N-terminus of the construct links to the cover slip surface, and then the biotin links to a streptavidin-coated magnetic bead. Using a magnetic tweezers apparatus, we can pull on this structure and then measure the lifetime of the ICD to PDZ bond as a function of force. I'll mention in passing that if any of you are interested in doing single molecule biophysical measurements, magnetic tweezers can be constructed for a few $100s. And the data are readily and simply collected and analyzed. So this is a great way into the business. Now, what Whipple found was quite surprising and quite spectacular, actually, is that just like the cadherin catenin to actin bond, the bond between nectin-1 and the ephadin PDZ forms this monster catch bond with a lifetime that increases almost 40-fold in going from no force. This is measured under using biochemical techniques to about 6 piconewtons of load. Now, he repeated this for JAM-A and found that the difference in binding lifetimes was even more extreme. Now, this is totally shocking. PDZ domains have been studied extensively using sort of common biochemical approaches and also high throughput systems biology approaches. But to my knowledge, nobody had thought to check to see if they might have force sensitive properties. Now, at this point, it might be useful to talk about how force sensing can occur. Now, I'll just remind you of some physical chemistry that you might recall with more or less fondness, in that the rate of a reaction scales with some energy divided by kBt. Now here, that energy I'm replacing with a force times a distance. This is mechanical work. And I'm saying that the observed rate, Kf, scales like the rate at 0 force times this exponential. Graphically, what that means is that this mechanical work term is added to the energy landscape for the protein. Here's a protein, for example, that exists in either compact or extended states, and that as you pull, you stabilize the extended state. Now, what this means is that force can continuously tune this equilibrium. What is a force to a protein? Well, thermal energy is about 4 piconewton nanometers. 1 ATP is the equivalent of 80 piconewton nanometers, and 1 myosin, depending on who you ask, produces about 2 piconewtons. So these PDZ catch bonds are responding to a couple myosins worth of force. Now, we have analyzed the data as best we can, and at the moment, the data fit best to the simplest possible catch bond model, where the bound state can dissociate via one of 2 pathways. In one pathway, in the absence of force, unbinding is fast, and over in one direction, and then slower in the second direction along this reaction coordinate. Now, what happens when we add load is that this, just as I showed before, tilts the energy landscape. This slows down the prevalent dissociation pathway, but speeds up the force-dependent pathway. At intermediate forces, though, the effective force is overall to slow down dissociation. We've also been lucky to collaborate with Abhishek Singroy's group at ASU, who has done cutting-edge molecular dynamics simulations to examine how this might work in a molecular sense. What his work shows is that as you pull on the complex, the PDZ domain actually begins to clamp down around the peptide ligand. So what we see is that for many of these contacts, the amount of solvent accessible surface area decreases, meaning that it becomes more stabilized as you pull. And what's kind of special, I'll just advertise his approach, is that the simulation comes very close to predicting equilibrium conformations in free energies as you pull, which is technically quite demanding, and for us has been revelatory. So just for example, here are data showing that as you pull the peptide ligand out of the pocket, you actually, just as I was showing previously, get a slight decrease in the solvent accessible surface area, which again is predicted to stabilize the complex. Now, it is reasonable to wonder if everything that my group measures is just a catch bond, right? And reassuringly, the answer is no. So here are data for the dissociation of Z01, PDZ3, and the same JAM-A cytosolic or intracellular domain. And what we see is that instead of forming a catch bond, this interaction pair forms what's called a slip bond. In other words, a bond where the harder you pull, the faster the dissociation occurs. Now, this is expected behavior, but what's less expected is the extreme force sensitivity of this interaction. So what we're seeing is that force of even 1 myosin molecule, shown here, accelerates the dissociation by nearly 2 orders of magnitude. This is among the most force-sensitive bonds described in the literature, and certainly the most force-sensitive bond that my lab has measured. Now, when you put these two together, you see something interesting. Remember, I showed you that JAM-A binds to both ephedrine and to ZO1. And if we graph the data on the same scale, we see that the dissociation rates actually flip at about 2 piconewtons of force. The idea being that JAM-A would prefer to bind to ZO1 at 0 force, but to ephedrine at higher force. Now, we were still quite curious about exploring the consequences of this observation. So here are two ideas. The first is that just like the cadherin-catenin complex, our data suggests that these PDZ-dependent interactions can reinforce cell-cell junctions to protect cells from mechanical stresses. What we're extremely interested in exploring, though, is that mechanical force can act as an input to help drive the self-organization of these very complex molecular assemblies. Now, the first taste of this may be the observation that I just showed you, which is that force can help drive the sorting, potentially, of JAM-A between its two possible ligands, ZO1 and ephadin. And ongoing work aims to explore the role of force in the self-organization of these vertically stratified cell-cell junctions. Now, before I transition to the next part of my talk, I want to zoom out just a little bit. So I mentioned that PDZ domains are prevalent. They're actually all over cell biology. Here are only proteins that exist at cell-cell junctions, my preferred domain of exploration. And you can see that they are just riddled with PDZ domains. Almost all of these are almost completely, either completely or almost completely unexplored at either a biochemical or biophysical level. So we're very curious about this. The consequences of the PDZ-based mechanosensing and more broadly, mechanosensing by these scaffolding proteins are only beginning to be explored. What will be necessary in order to do this is the development of tools that allow us to measure not just one protein ligand interaction at a time, but hundreds or even thousands. In other words, the development of a systems approach to mechanobiology. This is a challenge that we're very much looking forward to approaching. Now, for the next part of my talk, I want to shift gears, talk about another cell-cell adhesion protein, the complex that is necessary to generate organized polarized tissues. But because I'm a lazy PI, this involves shifting between PowerPoints, presentations with different slide formats. So if I'm really smooth, this might be relatively quick. If it's more typical, it may take me a few tries. All right, do you all see a flywing? Yeah, okay. So the next thing I'd like to tell you about is a problem called planar cell polarity. Planar cell polarity makes the little hairs on this flywing all point in the same direction, but as I'll tell you in a few slides, it is also essential to make a human. And I'd like to focus your attention on just how cool this problem actually is. So somehow or other, hold on while I get my pointer going. There we are. Somehow or other, these cells over here know to point in the same direction as these cells over here. How do they do that? It's a good problem, and it's one that I've been lucky to get to work on for the last couple of years. Now, just to describe what planar cell polarity is a little bit better, imagine each one of these cans is a cell. Up, down, as I mentioned earlier, is apical basal polarity. And then a perpendicular axis in plane polarity defines proximal distal. Oh, are we not seeing the correct? Yes, we must not be seeing the correct screen. Are we okay?
Háblale.
Let's see.
We are seeing the presentation.
Okay, great, cool. Yeah, then I will continue.
Continue.
Yes. All right. So for the talk, I'll be describing the cell orientation in terms of proximal distal, which is the in-plane polarization of these Pepsi cans with all of their tabs pointed in the same direction. Now, I mentioned earlier that this is necessary in order to make a human. So just for example, here's a simple example. Planar cell polarity helps align all of the hairs on this mouse or on you, actually. So here's a mutant in planar cell polarity signaling, and you see these swirls. Now, more serious is that planar cell polarity also helps define the proper organization of the internal organs. So here's a mutation called mutations in PCP lead to situs inversus. And they also lead to quite serious birth defects. For instance, those that lead to failures in neural tube closure that lead to spina bifida, which occurs in approximately 4 in 10,000 births. So this is quite a prevalent source of birth defects. Mutations in PCP signaling also lead to a number of different congenital heart diseases that occur in up to 75 births out of 1,000, which can, in some cases, have quite serious health consequences. Now, the point being is that you cannot build a person without planar cell polarity. Now, let me describe PCP signaling in just a little more detail. So, two cells, cell 1 and cell 2, the communication between these two cells is mediated by 5 proteins that we'll talk about today. On the proximal side of cells, Van Gogh or Vang and Prickle. On the distal side of cells, disheveled and frizzled. These two subcomplexes are linked by flamingo, which is a transmembrane cadherin that links these two cells together. It is not the topic of today's talk, but we know from indirect evidence that frizzled and Van Gogh affect the conformation of flamingo, and that in this way, the distal and proximal complexes indirectly communicate with each other. How this occurs in a molecular sense is still not understood. What I want to talk about today is how the overall polarization occurs, though. So how, in other words, do you get proximal and distal to polarize such that you get the right stuff here and here? And how do you, in turn, prevent the incorrect orientation? All right. And as you can see, I'm speeding up a little bit so that you all can get to your lunch on time. Now, something that had been noticed for a long time, but that had not been adequately explored, is that these proteins are not homogeneously distributed along the membrane, but assemble into clusters, just like the TCR clusters that we heard about in the previous talk. And that if we look along one cell-cell junction, we see, for example, that Van Gogh is enriched in clusters, and that these clusters, as expected, have mostly proximal Van Gogh. Now, interestingly, No previous study, to our knowledge, had examined the role of cluster formation in PCP signaling. So this motivated the postdoc who worked on this, Celis, to ask two questions. First, what is the role of cluster formation in PCP signaling? And second, which of the many possible forms of feedback between these 5 protein components are required for proper polarization to occur? Now, tackling that first question, there are various physical mechanisms for cluster formation that have been described in the literature and that are in theory possible. Clusters could form of roughly uniform size. You could see a coarsening where the big clusters gobble up the little clusters. This leads to a bimodal distribution of cluster sizes. And then in other systems, for example, in classical coherence, you see that a combination of fusion and fission leads to power law distributed cluster sizes. As we'll see in a moment, examining the mechanism of clustering in the system can provide us insight into how polarization occurs. Now, excuse me. In order to figure out the size distribution of these clusters, Celis used a quantitative photobleaching experiment. Here is the wing of a pupal Drosophila embryo or fly. And these little speckles are clusters. And what we'll do, is watch the intensity at each cluster as we deliberately photobleach the wing. So when we do that, we see that towards the tail end of that bleaching curve, we see quantized steps. And by quantifying the size of those steps, we can infer the average fluorescence intensity from 1, 2, or 3 EGFP molecules. We can then extrapolate back to the beginning of the movie in order to figure out how many GFP tagged Van Gogh, for example, molecules were in that cluster. And this cluster, for example, had 8. And Celis has done this for, at this point, hundreds of thousands of clusters across many flies, and for all of the components of the PCP cluster that I've told you about. Now, the other trick is that using fly genetics, it is possible to make patches in the wing where some cells express EGFP tagged VANG and some do not. That means that if we look at the borders of these patches, we can define the proximal sides of cell-cell junctions and the distal sides of cell-cell junctions. Now, with all this information in hand, or with this technique in hand, we can begin to see how these clusters are built. At the bottom row will be the proximal clusters, top row will be distal clusters. For flamingo, which bridges across a cell-cell junction, we see that cluster sizes are exponentially distributed. So this is classic science where we had, remember, 3 possible models for how these cluster sizes should be distributed, and the answer is none of them. So this was a surprise, and we had to think about it for quite some time. Reassuring, and I should say before I move on, that this is a semi-log plot, and it shows the fraction of clusters bigger than some size. So that, for example, all of the clusters are bigger than 0, and none of the clusters are bigger than, say, 35. The thick line is a fit to a single exponential, as shown here. The mean, or cluster size, is, in this case, 4. So these are small. These are much smaller than the lat clusters. Reassuringly, when we look at flamingo on the distal side, we get the exact same average mean size. Each one of these lines, I should say, is data from a single fly wing. So this is nice. Flamingo is, on average, symmetrical, exactly as one might expect. Now we can immediately say that we have ruled out all of these models for how these clusters assemble. Cool, we have something new to learn. Now, I'll just note that before we go on, the exponential distribution in cluster sizes implies that the binding and release rates of individual components are independent of cluster size. This provides a strong constraint on any model of how symmetry breaking occurs and is a property that we'll exploit in a few minutes. Now, next, we'll look at incorrectly positioned frizzled. So this is proximal frizzled, shouldn't be there, but we do see a few. And distal prickled, also shouldn't be there, but we do see a few. That will also become useful. As expected, we see an enrichment of distal frizzled and proximal prickled. Just like flamingo, the size distributions here are also adequately described by single exponentials. So what we've learned from this is that frizzled and prickle are imperfectly polarized, and that the degree of imperfection is, let's say, on the order of 5 or 6 to 1. So the system is not perfect. Now, finally, we'll look at Van Gogh. We see that in contrast to these molecules, which occur in a roughly 1-to-1 stoichiometry with flamingo, we see a large excess of proximal Van Gogh. And we see an appreciable amount of distal Van Gogh. This was a surprise in that Van Gogh had previously been thought to occur only on the proximal side of cell-cell junctions. So we now have a large amount of data that can help constrain models for how polarization occurs. Now, what Silas next wanted to do was to understand if the average size of clusters might matter. And in order to specifically probe this question, he took advantage of mutations in a distal scaffolding protein, disheveled, that limit its ability to oligomerize. So disheveled oligomerizes via what's called a Dix domain, which makes a beautiful helical coiled coil polymer by itself. Mutations in the Dix domain that limit the ability to polymerize also limit its function, shown here in a Wnt signaling assay. So I neglected to mention it, but frizzled and disheveled are also core components of the Wnt signal transduction pathway. So using these mutations, we can see if the oligomerization of disheveled has any effect or any importance in planar cell polarity signaling. So here are data for a wild type fly wing, and here's the distribution of Van Gogh cluster sizes. And then the same experiment for a series of mutations that progressively decrease the ability of disheveled to oligomerize. And we see that the stronger the mutant, the smaller the overall Van Gogh cluster size, and the more disturbed polarity at the wing is. What this tells us then is that the decrease in cluster size results in a decrease in the fidelity of polarization. Now, one more bit of data, which is that in very recent work, Silas has been able to measure the size distributions, roughly, for 2 components of these clusters at once. So I'm showing you here the average, the brightness as opposed to the molecular counts for Van Gogh and frizzled in the same cluster. And what we see is that these roughly scale with each other. So there's some noise in the measurement, and There's also some noise in the biology. We saw that earlier, but that in general, more frizzled implies more vang. This is expected. Now we can do a little bit more sophisticated measurement where we look at distal prickled. This is prickle on the wrong side versus proximal vang. This is vang on the correct side. And what we see is that on average, we see incorrectly oriented prickle only in the smallest clusters, and that large clusters defined by proximal vang have little or no distal prickle in them. We can do a similar measurement with vang and frizzled. So just as I showed you before, clusters that have vang on the wrong side, on the distal side, are generally small. Clusters that have lots of frizzled on the correct side generally have little or no distal vang. So what this tells us is that clusters with incorrectly oriented prickle or vang don't grow. Now we are still building this out, but we are beginning to use these data to develop quantitative models for how this polarization can occur. Now, we are not the first to do so. What I'll point out is that there are 5 protein components here. From biochemical and cell biological data, most of these components can bind to most of the other components. There are multiple phosphorylation and other post-translational modification events that I'm not telling you about. And this results in a vast set of possible activating and inhibiting interactions between these components. Because of this, it's been exceedingly difficult to develop predictive models for how PCP occurs. I don't intend to present you with a molecularly detailed model now. Instead, what Celus has done is strip this down to its barest essentials, where we have a distal complex labeled F for frizzle and a proximal complex labeled V for vang. He has found that the data can be modeled with only a few input parameters. A parameter alpha that controls cluster growth, a parameter gamma that couples the growth of a distal frizzled to a proximal vang. So the more proximal vang, the faster frizzled is added on the distal side. And then an inhibitory term where if there is incorrectly oriented vang, for example, it slows down the growth of frizzled and vice versa. So in other words, one activation across cell-cell junctions and one inhibition within the same subcomplex. Now, we then take these data, put them into kinetic Monte Carlo simulation, and we find that as desired, this model results in approximately exponential cluster size distributions. And again, this is consistent with our observation that the simplest possible growth law is sufficient to explain our data. It correctly predicts that so this is a bit of a simplification. Let's just say that in general, vang, which is mostly proximal, scales with frizzled, which is mostly distal. This is in accord with the data I showed you. But that incorrectly oriented distal vang occurs only in small clusters. That was also consistent with the data I showed you. And here they are, just to remind you. So this is a little bit less noisy. That difference may be due to experimental error, or it may be a failing of the model. We'll see. Now, what we're taking away from this measure is, from the model and our data, is that only weak intercellular inhibition and intercellular coupling are required for polarization. In other words, the system is quite robust in terms of successful polarization resulting from weak interactions. Now, remember we began with the question of how clustering is related to polarization. So here we're varying the on rate alpha. And we see that if the on rate is small, you of course don't get any polarization. But as if you include both a finite on rate and some relatively weak intercellular coupling, this gamma, you get this abrupt transition to highly efficient polarization. So, in other words, we find that polarization increases rapidly with cluster size, and that even weak, as I've mentioned several times, coupling across cell-cell junction is sufficient for polarization to occur. All right. Now, I am out of time. You all may be becoming interested in lunch. So I'll skip the conclusion slide and thank my research group. The first half of the talk was done by Vipul Vacharajani, who graduated relatively recently. That's him here. The second half is done by Silas Nissen, a joint postdoc in my group and in Jeff Axelrod's group, whose group I'm privileged to work with and who does all of the Drosophila wrangling that makes this study possible. I thank you all again for the opportunity to present some of our work, for paying attention to a Zoom talk, which I know is considerably less engaging than having somebody there in person. And I again apologize for my inability to be there with you all as I would otherwise have liked to be. If there's time, I'll be happy to answer any questions.
Okay.
Hello, Alex. A short question that could be relevant for here is you mentioned that you want to for you. There's a strong echoing. Wait for a second. Okay, so basically my question, which could be relevant for the audience present here, is you mentioned you want to study the junction mechanobiology systemic kind of way. What kind of technology you plan to have?
There are numbers of ways that a person could tackle this problem. The way that we're approaching it is we're midway through developing a high throughput microfluidics-based device that, if it's successful, should allow us to pull on many such complexes, as I've showed you, in parallel rather than one at a time. This is a big technical lift, but we believe it should be possible.
Okay, thank you very much.
Yes. Well, thank you.
Thank you for being here and for everything.
It was my pleasure.
Pero ok, bueno. Ahora es turno de la doctora Shira Landau. Ella es investigadora postdoc del Instituto de Biomateriales e Ingeniería. Esto en el Milica Radisic, un grupo de investigación líder en ingeniería de parches cardíacos, biomateriales, inyectables y sistemas microfabricados para cultivo celular. En su etapa postdoctoral, la doctora Landau continúa aplicando su experiencia en ingeniería de tejidos y regeneración vascular, contribuyendo significativamente al desarrollo de soluciones avanzadas en medicina regenerativa y biomateriales. Thank you very much for being here today with us. Now, I give you the word.
Yeah, hi everyone. Let me just share. Oh. Yeah, can you see my screen?
Sorry, I think we can't hear you.
Oh, can you hear me now?
Yeah, I don't know.
Can you hear me?
Yes.
Okay, I'll just get closer to my computer. Can you see my screen?
Yes, we can see it.
Okay, great. Okay, let me just. Okay, hi everyone. I'm Shira from the Radisek Lab, and I'm here on behalf of Professor Melissa Radisek, who couldn't attend. And today I'm going to talk about vascularized cardiac organ on a chip systems for studying function, disease mechanism, and treatment strategies. So cardiovascular disease is the leading cause of death worldwide, with over 20.5 million cardiac vascular disease related death in 2021. The regenerative capacity of the heart is extremely limited, and suitable donors' hearts for transplant cannot meet the demand and require recipients to take immunosuppressants for life. So a way to overcome this is use in vitro cardiac models, and they can enhance cardiovascular disease treatments by using it for personalized disease modeling, for drug testing, and for even organ regeneration and implantation. We can take microtissues and implant them into the heart. So current models have been developed over the years to create cardiac models, such as cardiac sheets, spheroids, patches, stripes, rings, tubes, and even models of ventricle cavities. But what's common to all of these is that they're mainly composed of cardiomyocytes, whereas the heart is composed of other cells, as you can see here in this map. And today I'm going to talk about incorporating fibroblasts, endothelial cells, myeloid cells, specifically macrophages, and cardiomyocytes into these cardiac tissues. So the outline of my talk is this. I'll start by talking about development of vascularized tissues in vitro, and then I'll talk about vascularizing the cardiac tissue, and then developing methods to enhance tissue 3D printing. So for the first part of my talk, it's extremely important to vascularize engineered tissues. We know that the viability and the clinical success of the implant-built tissues depends highly on the integration of the tissue, and it needs oxygen and nutrient to penetrate into the implanted tissue in order for it to integrate. And here you can see in this video, implanted vascularized tissue, and the vascularized tissue is in green, and it's connected anastomosis to the host vasculature in blue, and you can see rhodomine dextran is flowing through the host mice vasculature into the engineered vasculature. So how do we create this engineered blood vessels? We do that by co-culturing stromal cells, a different kind of stromal cells with endothelial cells. And these cells will spontaneously form blood vessels. As can be seen in this time-lapse video, you can see the blood vessels in red, and the green are just stromal cells. So why do we need these stromal cells? They'll provide structural support, regulation of blood flow, angiogenesis. They provide barrier function, and they provide inflammatory response under pathological conditions. We can use various types of supporting cells. In the literature, you can find fibroblasts, dental pulp stem cells, smooth muscle cells, parasites, and mesenchyma stem cells. You can also control the in vitro blood vessel formation by using either mechanical geometry, matrix geometry, and composition, and by applying mechanical forces. So here is an example of how we played with the composition of the matrix by collaborating with Professor Tony Weiss from the University of Sydney. We coated polymeric scaffolds, in this case PLA, PLGA, with tropoelastin, which is elastin precursor, and is known to adhere to integrins within the endothelial cells. And this resulted in an increase in blood vessel formation. You can see that untreated controls have a lot less vasculature compared to the tropoelastin treated scaffolds. And in vivo, you can see that this also resulted in higher vessel penetration after 10 days. We can also apply mechanical forces onto our scaffolds. So here, specifically, we applied a stretch in our scaffold. So we have 3 different conditions. We have the free-floating scaffold, static stretch scaffold, and cyclic stretch. And you can see that the vessel oriented differently. So free-floating, as expected, you can see randomly oriented blood vessels. Under static stretch, you can see that the vessel oriented parallel to the stretching direction. And under cyclic stretch, you can see that the cells orient perpendicular to the stretching direction. And this is dynamic. You can start with static stretch, where the cells will align in parallel to the stretching direction. And then when you start to apply cyclic stretch, the cells will reorient themselves. It reveals a competition between the 2 mechanosensitive mechanisms. We also saw that the applying cyclic stretch results in stabilization of the vessels. We get vessels that are stabilized over longer periods of times, and this enhanced secretion of pro-angiogenic cytokines, as can be seen here with AGF, VGF, and one higher secretion within scaffolds that were under cyclic stretch. Interestingly, when we compare 2 different types of endothelial cells, we have lymphatic vessels that exhibit different response to cyclic stretch compared to blood vessels. So here are lymphatic vessels, which are tagged in green. You can see that although we're talking about cyclic stretch, the vessel oriented in parallel to the stretching direction, whereas blood endothelial cells tagged in red here align perpendicular to the stretching direction. So we have 2 different endothelial cell types and 2 different responses to the mechanism, to the mechanical forces. So now I'm going to talk about vascularizing the cardiac tissue. So why do we need to vascularize the cardiac tissue? Endothelial cells have a very important role in the cardiac function, regulation, and maintenance. It has a strong effect on cardiovascular and on cardiomyocytes phenotype and survival. Endothelial cells are known to secrete factors such as nitric oxide, endothelin, and they have a very strong effect on the overall cardiac metabolism, growth, and development. So there have been attempts to create vascularized cardiac tissues, but either their physiological relevance is pretty low, we don't have any blood flow within them, or we can induce flow, but then it's low throughput. So first we wanted to create blood vessels, as I showed you before. We started with looking at the effect of stromal cells, and again, without stromal cells, you can see that you barely see any blood vessel network formation, whereas when we add stromal cells, we get this vessel stabilization over 2 weeks. And you can see also that when we add the dead pulp stem cells, in this case, that these are the stromal cells, we get collagen 4 secretion around the vessels and alpha SMA positive expressing stromal cells around, meaning the vessels are more stable. We also saw with bulk RNA sequencing, as expected, there are more collagen secretion in the presence of stromal cells. And then we added cardiomyocytes into our tissues. And interestingly, we observed vessel degradation when cardiomyocytes were added to the tissue. And here we saw in the gradient experiment that the more cardiomyocytes we add, the more vessel degradation we get. And we see that the beating amplitude is getting stronger as expected. With more cardiomyocytes, you have stronger beating. So to overcome this, we decided to incorporate another cell population. So cardiac resident macrophages are known to be CCR2 negative, and they're known to suppress inflammatory responses following acute myocardial injury. They're known to be required for the heart's regenerative capacity, promoting scarless repair, angiogenesis, and cardiomyocyte proliferation, and they're physically localized near endothelial cells and composed of angiogenesis-enriched genes that enhance endothelial vessel formation. They interact with smooth muscle cells to stabilize arterial smooth muscle. So in collaboration with Professor Gordon Keller and Professor Slava Eppelman from U of T, we use primitive-like macrophages, which are embryonic stem cell derived, and they're live-1 positive and CCR2 negative, similarly to cardiac resident macrophages. And here you can see that when we looked at the genes, their genes are mostly expressing cardiac resident macrophages and not expressing monocyte markers. So our goal was, first of all, to determine the reciprocal effect of the different cells, and then we incorporate them into the BioWire2 system to test their contribution to cardiac tissue development. And the third aim is to incorporate these cells into the perfusible iFlow plate system to enable formation of functional vessels. So first, we mixed all these cells together into this fibrin hydrogel. We have endothelial cells, dental pulp stem cells serving as stromal cells, cardiomyocytes, and macrophages. And here you can see macrophages are tagged in red. You can see that without macrophages over time, we get this vessel degradation. And with macrophages, you can see that some vessel stabilization occurred. We see that the macrophages interact physically with the vasculature. We found them wrapping around the vessels, bridging in between vessels, and to confirm that this physical interaction is needed, we use this transwell experiment where in one side of the well, we seeded a tissue of endothelial cells, dental pulp cells, and cardiomyocytes, and then the other side of the well, we seeded the macrophages, and we saw that the effect is reduced, leading us to conclude that their physical interaction is needed. Then we looked at the cardiac function, and we noticed, even though we're looking only on a small fibrin tissue, that with incorporation of macrophages, we got enhancement of cardiac functionality, and they're having a better response to electrical stimulation. You can appreciate the strong beating of endothelial cells and vasculature and macrophages around in this tissue. And to further quantify the way they're beating, and we use the BioWire platform, which allows us to detect the contractile forces of the cardiac tissue by measuring the two elastic wires here that are deflecting, and then we can measure their movements, and from that exert the forces. And we seeded the same cell combination in the endothelial cells, macrophages, dental pulp stem cells, cardiomyocytes into the BioWire system. As similar to what we saw before, without the macrophages, we get these clusters of endothelial cells. And with macrophages, we get nicely forming branch blood vessel network. So in terms of cardiac function in the system, we can see how with macrophages, the tissue is nicely compact and it's strong and it's beating more stronger. And you can see that in terms of cardiac function, excitation threshold was reduced, and maximum capture rate is increased, and active force is also increased, leading us to conclude that the macrophages also help in cardiac function. To further analyze the vessel function, we use the iFlow plate system, which allows us to create functional blood vessels that have flow within them. So the way it looks, it's a 384 well plate. Every 3 wells are a unit, and they're connected through channels here. So we have the inlet, the outlet, and the main tissue where the vessels are forming. And similar to what we had, so again, endothelial cells, DPSCs, cardiomyocytes, and macrophages. So without macrophages, we have this disconnected vasculature that didn't allow the flow, rhodomine, dextran, and polystyrene beads to flow through the channel. Whereas when we added macrophages to our system, we saw that the rhodomine, dextran is flowing nicely within the vessel network. And also polystyrene beads, as can be seen in this video, are flowing nicely within the network. So to understand what's causing these macrophages to have this strong benefit on both the cardiac tissue and the vessel tissue, we looked at the media, the secreted media, and then we looked at mitochondrial DNA within the media. So the mitochondrial DNA, which is known to be cytotoxic, is reduced in the presence, ND1 and ND2 is reduced in the presence of macrophages, whereas LDH is also reduced. So overall, a lot less cell stress. And when we looked at cytokines that are secreted, we get this MMP, which is obvious because we're adding macrophages, but we see some increase in proangiogenic factors as well. And when we looked at bulk RNA sequencing, we saw that there is an increase of enriched pathway that related to cardiac function, and also an increase in collagen 4, which are basement membrane proteins, and increase in MMPs as expected. To further understand the mechanism, since in bulk RNA sequencing, we can detect the individual effect of each cell. We performed single nucleus RNA sequencing, and here you can see that we detected 8 subpopulations within our tissues. So we have endothelial cells, DPSCs, as expected, but we also have fibroblasts, which are known to be residual cells within our cardiomyocyte differentiation. We have macrophages and 2 subpopulations of cardiomyocytes. So we have more mature cardiomyocytes, which express MYH7, and less mature cardiomyocytes, which express only MYH6, and 2 unclassified cells that are probably residuals of the impurity of our cell differentiations. So when we looked at the angiogenesis-related pathway within the every cell type, we saw that there's a significant increase within the stromal cells of both angiogenesis pathway score and VEGF signaling pathway, leading us to conclude that the macrophages induce the stromal cells to become more pro-angiogenic. When we looked at single nucleus RNA sequencing combined with secretomics analysis, we got this set of genes, and then we looked specifically at these genes, and we saw that they're mostly related to cell adhesion, migration, ECM, and basement membrane, also VEGF signaling. And again, when we looked at the specific cell genes that are expressed within these the specific cell population, we see that they're mostly expressed by the stromal cells. Again, macrophages induce the stromal cells to become more pro-angiogenic. So just to summarize this part, incorporated cardiomyocytes in vascularized tissues lead to blood vessel destabilization. Macrophages help to restore and stabilize vascularization and enhance cardiac tissue function. Our results demonstrate that primitive macrophages enhance both cardiac and vascular performance via two primary mechanisms. So there's a direct cellular interaction and through the release of MMPs alongside with pro-angiogenic and pro-cardiac cytokines, mainly mediated through stromal cells. So next, we're aiming to develop a platform that will allow us to assess both cardiac function and vessel function. And then the biological side is to understand more the underlying mechanism of the macrophages function through inhibition studies. So the last part of my talk is developing methods to enhance tissue 3D printing. And we're doing that by incorporating elastomeric particles into bioinks to enhance 3D printed tissue stability. So 3D printing limitation. It's impossible to print polymers with cells because organic solvent toxicity or the requirement of high temperature during the printing. And we all know that hydrogel lack the mechanical support that results in the collapse of the structure. So we have this really nicely printed structure, but over time, the cells will compact. And at day 7, you can barely see the initial structure. So our aim was to incorporate something that will help in stabilizing the hydrogel cell structure. And we use POMAC, which is a polyester, and it's photo cross-linkable. And we create these particles, combine them with cells, put them in a hydrogel, and then you can use two different any printing type that you want. We tested here extrusion printer or stereolithography printer, and we overall got enhanced mechanical properties. So the way we make it is by either suspension polymerization. So we have a needle going into searing bath of PVA solution, and and then the dropping particles will spontaneously form these particles, and then we UV shine it, and the particles are cross-linked, and you can see that we get these particles with multiple sizes. We can also use microfluidic particle synthesis, and in this method, we get monodispersed particles. So we then combine these particles within hydrogel, and here we used PEGDA and GELMA, and we got less, and with PEGDA, we got decrease in the swelling of our scaffold, and with GELMA, we got an increase of the Young modulus, and you can appreciate how elastic and stable they are when we tested them both with tweezers and compressing. So the transparent scaffold is without the particles, whereas the white one is with particles. So the scaffold that was with particles, we can compress it multiple times and the structure maintained. A similar thing happened when we use tweezers, that only GELMA are easily breaking, and GELMA with particles are very stable. You can see it also here in the mechanical properties, higher Young modulus, and also higher ultimate compression strength. stress. Then we added cells into our scaffold. So in this case, we wanted to create blood vessels. So we use the same cell combination of the TL cells and the pulp stem cells with formal particles into a fibrin hydrogel or gelma hydrogel. And we saw that the particles are really seeing, the vessels are really seeing the particles as anchor points. And within time, we got more vessel stabilization. The vessels are anchoring to the particles. We also seeded cardiomyocytes into our heart-shaped scaffolds, and we saw that there are more cells that are adhering to the scaffolds. We hypothesized that this is because without particles, we have this hydrogel that is very smooth, and with particles, we get this roughness that the cells like and adhere more. So, and then this results in overall increased mechanical properties of our cardiac tissue. So excitation threshold is reduced, maximum capture rate is increased, and the amplitude of the beating is stronger, as can be seen in these videos. Then we implanted these scaffolds into mice, and we also got, we saw that structure is more maintained with the particles, so they're easily breaking when we don't add the particles, whereas with particles, we get better stable structures. We also noticed that we get more vessel penetration within with and without cells, the scaffolds that are implanted into mice. And we also get the penetration of macrophages, both CD68 and pro-regenerative macrophage marker CD206 is increased when we have the particles in our tissue. And yeah, by that, I would like to thank my amazing supervisor, Professor Milica Radisek, all the lab members that helped me, and yeah, thank you for listening.
For the presentation. Are there any questions?
No. Now can you hear us?
Yeah, I can hear you now.
Yeah, thanks. First of all, thank you for this beautiful talk. I'm Tatiana from the Faculty of Science, and we have two students who want to ask.
Hello, my name is Diana. I have a question about the method. When you add the endothelial and stem cell, do you add any growth factor?
So we use which part of the talk are you referring to, with the cardiac tissue or only endothelial cells and stromal cells?
For endothelial and stem cell.
Okay, so basically we use EGM2 media, so it contains pro-angiogenic factors, and when we use the cardiomyocytes and macrophages, we combine EGM2 media and StemPro. media together. So they're both commercial media that have cytokines within it.
I think that what she wants to ask is when you put the endothelial cells in order to have like a tube, which is a capillary or something like that, did you put something, some factors to induce the organization of the cells, or these cells just get organized by the... Sorry, because the microphone is a little
Yeah,
bit
yeah, no,
strange.
no, I understand. It's okay. Yeah, so we use EGM2 media. It's commercial media. They don't really say what it contains, but we know that it contains pro-androgenic growth factors. So yes, that the answer is yes, we have it. We just don't manually use it. We have we get like pooled cytokines within the media that we use. Yeah.
And then first you put these cells and then the other ones in like different timing or all at the same time?
Yeah, we mix all the cells together in the same time. Yeah.
Okay. And we have another
Hi. When you refer to scaffold, are you referring only to Matrigel or did you do other tests?
Oh, which scaffold?
Así que le quieres preguntar que hablo de los scaffolds y todo lo Matrigel, ¿no?
Yes.
No, no te entendí.
So are you asking what kind of scaffold did I use?
Oh, when you refer to scaffold, are you referring only to Matrigel or do you do other tests about the scaffold?
Other scaffolds.
So we use fibrin as a scaffold. We mix the cells with fibrin in the macrophages part.
Thank you.
Is there another question? No? Okay, thank you very much for being here today and taking the time to be with us. We are very honored to have you here. So thank you very much.
Yeah, thank you.
¿Cómo se apaga esto?
Sesión de la mañana. Recuerden, hasta el viernes, todas las mañanas, allá afuera está el programa. Tendremos las pláticas y después los que se registraron para la parte práctica. Haremos la parte práctica en el Lansbiodit, en el edificio de física biomédica en el auditorio. Digo, en el auditorio, en donde está el estacionamiento de alumnos en el segundo piso, ahí los vemos. Pero tenemos la sesión de pósters después de la comida, que es aquí afuera, están aquí. Ahorita tenemos un espacio de comida de 1 hora y los esperamos de regreso para ver los pósters y luego ir a la parte práctica. Y desde luego los esperamos mañana para las siguientes pláticas. Muchas gracias.